Hello everyone, my name is Soum. Today I'm very happy to give a presentation about my research topic. My research topic is quantitatively controlled fluidic flow with ballpoint pen printed pattern for programmable photo paper based microfluidic device. To begin my presentation, I would like to give a clear answer to why microfluidic devices have been intensively developed by many research groups. First, if you look at the top picture, you can see this is the tool that we use in the lab to handling solution in the milliliter scale or liter scale. This means that you always use large volume of solution of chemical for your experiment. Recently, many research groups have been developed the small-scale device called microfluidic device is designed and fabricated to handling microliter scale of the sample, which is you can mixing two reagents together on the single sheets of paper. You can transport your chemical reaction to the specific area on the paper through the microfluidic channel. As you know, many chemical reactions happen when we mix together. That's why this paper microfluidic device or microfluidic device have so many applications. First is a lab-on-chip application. Lab-on-chip application, you can string the entirely analytical lab into very portable device that work as almost the same thing in very small scale. You can use to analyze chemical or biological sample in the small area here. Based on this lab-on-chip application concept for analytical, chemical, or biological sample, many researchers have developed the very simple and low-cost sensor, which is made by microfluidic device on paper. As you can see here, we can detect glucose by using colorimetric method. We can detect protein, but the size of the sensor is very small, just the size of your finger, and the cost is very cheap comparing to the big lab. You may use the commercial available strip sensor to detect the outbreak disease or some disease for drug screening that you may spend only 5 to 15 minutes to get the result out. It's a very interesting device. But to detect those chemical or biological sample, it's not very easy. Why it's not easy? Because you need to have the protocol. Some detection protocol is very straightforward because it just requires only a single step. It means that you put your chemical reaction on the microfluidic device, then you drop the sample, then you just wait some time and then you can get the signal, which is a color signal or electrochemical signal. But the other is not easy. It needs a lot of time, a lot of process. For example, like enzyming immunosorbent assay, or we call in short ELISA, it needs about 5 steps to get the signal out. Then, for example, if you need to detect 20 samples, it means you need to repeat 100 times of the process. So we want to make this process automatically, but not using the machine outside. If you're using the machine outside, the device it should be not portable and maybe it's more expensive. So that's why in our lab, we try to make the system, try to make the small device portable that can bring the sample, that can deliver the sample sequentially to the area that we want. So in this case, we want to control the fluidic flow in the paper-based microfluidic device without using external control system. When we develop those small-scale and portable devices, we will use those devices for various applications, including single-step assay and multiple-step assay with automatic process. So to fabricate those devices, we are mainly follow two protocols. One is to cut the channel. Of course, we can cut the channel using cutter, which is very small, portable, and low cost. Then we laminate those cut channel on the surface of our paper. Because paper is printable surface, we can modify surface of paper using other functional material, for example, hydrophilic material or hydrophobic material. In this case, we print silver nanoparticle ink, which is hydrophilic material, using ballpoint pen. And also, we can print hydrophobic material. The name in short as PTFE is hydrophobic material using the same tool, ballpoint pen with digital plotter. This pattern can be designed in electronic file and then upload into the plotter, and then we can print it. We can see silver nanoparticle pattern is a hydrophilic surface. It means have very high surface energy, that the contact angle is about 15 degrees, and the PTFE is very hydrophobic, which is very low surface energy. The surface contact angle is about 110 degrees. So we use these two functional patterns to modify the surface of the channel in order to control the fluid transportation in our microfluidic device. We want to increase the flow. To increase the flow is the way to make the flow faster is to make the channel become hydrophilic. So because our silver nanoparticle line is hydrophilic, then we modify silver nanoparticle line in our channel with different width. As you can see in this one as comparison between modify and without modify. Without modify, fluidic just flow like this. But with silver nanoparticle line, because the silver nanoparticle line have higher surface energy, and it tracks the liquid along its body, and then it makes the flow faster. As you can see here in this plot D, when you modify higher, like when you modify bigger without silver line, you can get the time delay shorter, it means flow faster comparing to the unmodified one. As you can see, if we increase more and more, we can see the velocity increase higher and higher. And this increasing is linearity increasing. You can see in this video, comparing between the modified silver line and without modified. You can see faster, very fast flow to here, comparing to the non-modified is still here. It means it takes longer time to reach to the end of the channel. This video is a real-time video. And we can increase the flow up to 15 times by modifying with the silver line. Increasing is not enough. We need more time, then we need to delay. So to get the high range of delay, we use hydrophobic pattern, use the same channel as the silver nanoparticle channel, as the channel which is we modified by the silver nanoparticle. But this time we modify hydrophobic pattern as a dot, not a line. But we increase the density of the dot in the channel. As you can see here, if we increase dot more and more, the flow distance time become longer and longer. It means the flow velocity become slower and slower. This is because after Teflon dot is a very low surface energy, hydrophobic. It doesn't want fluid to flow over it. And the dot actually act as an obstacle to resist the flow. That's why the flow becomes slower. You can see in this video comparing between the non-modified and modified, we can see the modified with more and more silver, a Teflon dot um, or hydrophobic dot. You can see it becomes slower. It takes more time. Combining between making faster and making slower, we actually can get high range of the controlling. We demonstrate just a Bruce concept design here that we claim that we can deliver 5 samples differently in sequentially using just a modification the surface. You may see number one, we increase the flow speed by modify the silver line here. And also number 5, we decrease the flow speed by modify a lot of dot here. So based on the time sequence here, we can see it's 60 seconds, the channel 1 deliver sample arrive to the main channel. And then the last one is channel number 5, the yellow color arrive the last. You may see in video movie here, you can see it's 10 times faster, and you can see it's sequentially delivered uh, to the main channel. In this case, mean we can mix the sample make the sample sequentially. For example, we want A and B react together, and then sorry, 1 and 2 react together, and then third number 3 react later, and number 4 and number 5, and so on. So this one we can control without any controlling system outside, just the chip itself, but we just modify the surface. So after making the device, we demonstrate the application, both single and multi-step reaction. So first, we just use a single-step microfluidic assay for detection of glucose using colorimetric method. I mean, we can detect glucose using the um, um, glucooxidase enzyme, and after reaction, it provides a color change. So in this case, we modify silver line in order to transfer or deliver sample very fast, so we can get early detection time and higher signal intensity. We can see here, when the time delay becomes shorter, the color intensity becomes higher. This one because of the time is very short, delay time is very short, and the reaction can happen early. So detect the glucose, which is an important parameter for diagnosis of diabetes. So this simple paper chip can detect the glucose concentration in your blood. Also, we demonstrate another type of application, which is multi-step, which we mentioned earlier. We can control the flow. We can get the sample delivery sequentially. This time, we did colorimetric detection of methylpyroxene. Methylpyroxene is a toxic chemical that people use as a chemical warfare simulant. So this can be detected by 2 steps. One is catalyzation to provide the P nitrophenol, and then the P-nitrophenol can deprotonate by strong base to provide the color that can be detected. So we designed this 2 channel like this. One is this deliver the MPO first, and then after that we deliver the strong base to protonate. By this protocol, we can detect the MPO from 5 micromolar and up. So this is the conclusion. Then we claim that we can fabricate it. Photo paper-based microfluidic device, which is the flow of fluid could be constantly controlled. And the flow can be increased 15 times and can be decreased 3 times by changing the material. And by variation the width and shape of the material, we can control the fluid flow quantitatively. So we also demonstrate the application by detecting the glucose and MPO successfully. This platform is not only for this tool of analyze for this not for detection of this tool, but they would be as a potential platform for various point of care testing device for detecting the disease. And if you have more concept, you can apply this platform for detecting other diseases like as HPV, HIV, or MERS or COVID-19. So this work has been published by Royal Society of Chemistry in Lab Ownership Journal, and this work has been selected as the inside back covers. Not only this work, I have a good opportunity to work on the other project, to work with other collaborator, and I have some notable contribution to the science and education community. And also, I have been present my work in various conferences. I'm very thankful to my supervisor, Professor Konushin, and my thesis referee, Professor Joe, Dr. Kwan, Professor Park, and Professor Kim. And also, I would like to thank to my lab member. Last but not least, I would like to thank to my collaborator, which has contributed a lot to my publication. Without them, I may not graduate this semester. Thank you very much.